Good afternoon. I'm Natalie. I'm Knowledge Exchange Manager for Horticulture at AHDB. Welcome to this webinar on tomato brown rugose fruit virus. Today we're hoping to provide you with a bit of an update on what we currently know about this virus and what plans are in place to mitigate for this threat to tomato and pepper production in the UK. During the webinar, there's the facility to ask questions. You can do this by typing into the question tab on the menu. We'll answer questions at the end of each of the sessions with Heiko and with Sharon. If you encounter any problems during the webinar, or you'd like to ask further questions after the webinar has finished, please feel free to email me on natalie.key@ahdb.org.uk. For information, this webinar is being recorded and will be made available to view on AHDB's Horticulture's YouTube channel. After the event, I'll put the link on the AHDB's dedicated tomato brown rugose fruit virus website alongside the other resources that are currently available for this. I will now pass over to Adrian Fox from Fera, who will provide us with an introduction to the virus and also host the remainder of the webinar. So we hope you enjoy it. Over to you, Adrian.
Good afternoon. As Natalie said, my name is Adrian Fox from Fera in York. And this afternoon, we're also joined by Heiko Ziebell from Braunschweig in Germany. And also with me here in York is Sharon Matthews-Berry from Defra. I thought we'd start with some introduction and background to the virus itself. So I'll take you through this, and then Heiko will spend some time giving some background to the problems that they've experienced in Germany with the virus over the last year, and then we'll finish with Sharon talking about the regulatory aspects for the UK. So I thought we'd start with some basics, bring us all up to speed. So to ask the question, what is a virus? Well, these are submicroscopic infectious agents. They have a very simple structure. At their simplest, they are simply some DNA or RNA genetic code, and that's surrounded by a protein code. Viruses reproduce within the cells of a living host, and they cannot reproduce outside of those cells. So they use the cell's own replication mechanisms to reproduce, and from that point forward, they generally go systemic within a host. There are around 5,000 recognized viruses, and there are many thousands being reported every year. And there are different viruses recorded in all species. So that's animal, fungal, and plant. They generally do not cross kingdoms, so you don't get animal viruses that infect plants, and you don't get plant viruses that infect humans. And very few viruses cause major problems. However, when they do, they can cause major economic damage, as we see quite often with plant viruses. So tomato brown rugose fruit virus is a tobamovirus, and again, just to give you some background to tobamoviruses, these were among the first plant viruses to be characterized. We know that they're seed transmitted at high levels in untreated seed, and once they're in a crop, mechanical transmission would be the main route. So that's either contact between plants or through plant handling activity, cutting, and so on. Tobamoviruses are highly stable, and they can persist for long periods of time outside of their host. And just to give you an idea of how stable some of the tobamoviruses are, they are viable in dried leaf material for over 10 years. Some work done in the 1960s looked at the sputum of chronic smokers, and that was shown to be contaminated with viable tobacco mosaic virus. And also pepper mild mosaic virus has been suggested as a marker of fecal pollution, or for water treatment efficacy, given that it will pass through the human body and will still be detectable in water courses afterwards. And there are several tobamoviruses linked to commercially damaging outbreaks in plants, such as tobacco and tomato mosaic virus in tomato crops. However, these are largely controlled through use of resistant varieties. If we think about tomato brown rugose fruit virus specifically, the reason that it's causing so much concern is that it can overcome the TMV resistance genes in tomato. It's spread through mechanical and contact, such as plant handling, and also recently bumblebee transmission has been demonstrated. Once it's present within a crop, it spreads rapidly, and I think we'll have Heiko to testify to that as we go through the afternoon. And also seed transmission is implicated, but not yet proven. Once plants are infected, they cannot be cured, and so prophylactic hygiene measures are used to try and minimise the risk of spread and to also limit the impact should an outbreak occur. And tomato brown rugose fruit virus has also been recorded on pepper crops. It's very much an emerging problem and only been known for around 5 years. So the first outbreak was recorded in Israel in 2014, and the virus is now present in all tomato growing regions of Israel. Later it was reported in Jordan. There have been outbreaks in Mexico and an outbreak in California. And there are also outbreaks in Europe, in Sicily and Germany, as we'll hear about later. And at present, we're investigating unconfirmed reports from both Belgium and the Netherlands, which, as I said, we don't have official reports for these, just anecdotal reports through the trade press. So a quick think about the symptoms, and one of the problems is that the symptoms on fruit can be very similar to mosaic virus. But the virus is named after the brown wrinkled rugose patches which can develop on affected fruit. Symptoms can vary with variety, and so you get mosaics such as chlorotic or pale patches on younger leaves and side shoots. Leaves can be crumpled and deformed or maybe narrowed, and you get brown necrotic streaks on stems. But as I said, the fruit can develop marbling symptoms which can appear similar to mosaic. But if you look on the AHDB website, there's a poster of symptoms and further information on the virus. And this, as Natalie said, is where the webinar will be signposted later. So I'll now pass over to Heiko in Germany, who will give us some background to the situation in Germany.
Good afternoon, I'm Heiko Ziebell. I work for the Julius Kühn-Institut in Germany, and we've been involved with some of the detection of tomato brown rugose fruit virus last year due to an outbreak in Germany. And now you should be able to see my slides. Is that correct, Adrian?
Yes, that's correct, Heiko.
Okay, perfect. So as Adrian has already said in the introduction, tomato brown rugose fruit virus belongs to the group of Tobamoviruses, and that group includes many viruses that infect commercially important crops like tobacco mosaic virus, tomato mosaic virus, cucumber green mottle mosaic virus, odontoglossum ring spot virus, just to name a few. And more importantly for us is that this new virus infects plants that are resistant to TMV and to TOMV. The described host plants are not only tomatoes, but bell pepper, petunia, solanum nigra, the black nightshade, tobacco species, and Chenopodium species. But the infection may not always cause visible symptoms, such in the case of petunia. So you might have an infected petunia, but you cannot tell that this plant is infected if you look at symptoms only.
Sorry to interrupt.
Oh, sorry.
Can you present your screen? I don't think we can see your presentation presented. Oh, we've lost it completely now.
Oh, I'm very sorry.
It is okay if you could just click.
Let us start again.
I think if you just go into the slideshow format. So if you go into the bottom corner and hit the...
No, I think it's just on the... Oh, there we go.
Yep.
Okay. So you might not be able to see visible symptoms in some of the host plants that can infect it with the virus. The virus was first found and reported in Jordan, but the outbreak in Israel was actually 1 year earlier, but it was only reported in 2017. And last year in August, we had the first reports from Germany, and that was the first description of this virus from Europe. And since then, Adrian also has said we found the virus in Sicily, California, and Mexico. In August last year, tomato growers in North Rhine-Westphalia were affected, and in particular, there were 7 growers that produced on more than 25 acres of greenhouse space tomatoes. And we had a rapid spread of infection, not only within the greenhouses, but also between production sites. The Julius Kühn-Institut is responsible for the pest risk analysis, and a high phytosanitary risk was identified with this virus, and therefore it was decided to treat tomato brown rugose fruit virus as quarantine pathogen with all the measurements that need to be taken in place. That means all the affected plant material had to be cleared and destroyed, and luckily it was towards the end of the tomato growing season, so the damages were not as bad as they could have been. All greenhouse spaces were disinfected, but that included also transportation boxes, tools, and so on and so on. And a couple of outreach activities were done. The Plant Protection Agency from North Rhine-Westphalia published some newsletters and articles with hands-on advice to the growers. And we are producing a fact sheet, which hopefully will be launched soon. There is a new series where we want to do little fact sheets for different diseases. So hopefully that will be published in the next few days. And we are also thinking of translating that into English if required. The symptoms can be quite unspecific or even unsymptomatic on leaves, but sometimes, as already Adrian said, you see a mosaic pattern. You can have smaller leaves, blistering. In some plant varieties, we see wilting and in the end plant death. The symptoms on fruits are more prominent with brownish and yellowish discoloration. But as I said, these symptoms can be variety dependent, and other viruses may cause similar symptoms, such as pepinomosaic virus or Physostegia chlorotic mottle virus. And most of you know that pepinomosaic virus is using a mild strain for cross protection. So many tomato plants within Europe are now infected on purpose with pepinomosaic virus. And at the time, it was quite unclear if those symptoms were caused by this cross protection induction or by a new virus. Just an example of the Physostegia chlorotic mottle virus, if you look at these tomato fruits, the symptoms are very similar to those induced by tomato brown rugose fruit virus. The transmission can occur by seeds and therefore also by young plants, and more importantly by mechanical means, and that involves all the tools, hands and handling, packaging boxes, knives, closed irrigation systems, and so on and so on. And recently it was also described that bumblebees used for pollination of tomato plants can transmit the virus within a greenhouse. And Adrian has the same resource as I. These particles are extremely stable, and even the sputum of smokers can contain viable particles of tobacco mosaic virus, and we therefore believe that this new tobamovirus has very similar properties and is also extremely stable. At the moment, we have 3 ways of diagnostics available: electron microscopy, RT-PCR, and ELISA. And I will talk a little bit in more detail about all these different routes. So electron microscopy can discover these virus particles in infected plant tissue. What I've shown here is a photograph from my colleague, Dr. Richard Pöggeler, and what you can see is the purified tomato brown rugose fruit virus particles. These are not samples taken from a plant. If you have a plant leaf, the detection might be even more difficult because you don't have that many particles available, or you have used not the correct leaf where maybe the particles are not present in high concentrations. However, electron microscopy is non-specific. We cannot distinguish by the look at this photograph whether this is tomato brown rugose fruit virus or TMV or any of the other tobamoviruses. If you have antisera available, you might be able to do an immunosorbent electron microscopy, thus fishing the virus particle out of infected leaf tissue, or you may be able to do a decoration, but this all depends on the antisera. And I will talk about the antisera in a couple of minutes. For diagnostics using RT-PCR at the moment, we recommend using group-specific tobamovirus primers, followed by sequencing. There are quite a few publications available with generic primers that can detect a whole range of tobamoviruses, and I've named a few here. But some of these primer pairs produce non-specific bands, even from healthy controls. We also tried the tomato brown rugose fruit virus specific primers from Luria et al., but those primers might not be detecting all the strain of this virus. So this is a comparison of two generic tobamovirus primers, and you can see here that the primer pair from Lee and al. Is able to detect infected plant tissue from tomatoes and tobaccos that were inoculated by us with the tomato brown fruit rugose virus. The healthy controls look fine. We do not get unspecific amplification, but if we use the primers that were published by Wolf Menzel et al., we do get some unspecific bands in the healthy controls, and of course that's a bit of a nightmare for a diagnostician because this is not a clear negative. And in any case, we suggest that you cut out these bands and send them for sequencing, because these assays do not tell you which of the many tobamoviruses is present in your sample. We also used a couple of other tobamoviruses with these primer pairs, and for us, the Lee et al. Primers worked quite well. They detected a whole range of tobamoviruses, while this primer pair failed to detect cucumber green mild mottle virus. And I spoke to Wolf, and he said that he designed these primers many years ago when there were only limited sequences available online, and therefore they might not detect all tobamoviruses. So if in doubt, I think it's always good to use a couple of different primers. Both work fine for the rugose virus, as I can show you here, but it depends on the diagnostic protocol that you are using. We started off out with a one-step RT-PCR protocol, and we knew that 3 samples named 17, 18, and 19 were definitely infected with the rugose fruit virus, but only 1 out of 3 samples were positive when we used these specific primers. None of the samples came up when we used the Lee et al. Primers, but when we repeated the protocol as a 2-step RT-PCR protocol with exactly the same primers, all 3 samples came up positive, and the likely explanation is that there is a denaturing step necessary at the very first beginning that we include in the 2-step protocol, but not in the 1-step protocol. And at the moment, we are using this as our first line of diagnostics and this protocol as the second line of confirmation test. At the moment, there's only a limited availability of anti-sera from us and the DSMZ, and the Israeli people have also produced their anti-sera against tomato brown rugose fruit virus, but as far as I know, it's not commercially available. The problem with anti-sera is that they are often cross-reacting with other tobamoviruses, so there is no specific detection of this particular virus possible, but it is suitable for mass screening of young plants, for example, and then if you find something, you need to confirm with other means that it's the rugose fruit virus or any of the other tobamoviruses. What can be done as prevention or as treatment? Adrian said treatment is not possible. Once a plant is infected, it stays infected and you can only destroy it. At the moment, no resistant tomato varieties are known, and we can only recommend that you use a very, very strict hygiene program in your facilities. Closely observe young plants, new young plants, or any other plant material. If you see any suspicious symptoms, contact your plant health advisor and test your plants if you suspect a virus outbreak. If the tomato brown rugose fruit virus infection is confirmed, all plant material needs to be destroyed and not composted. The tobamoviruses are not inactivated by composting. And all tools, tables, trays, greenhouse facilities need to be disinfected. In Germany, we have only one product that is licensed as a virucide that is licensed against for the treatment against TMV. And at the moment, there are no data available if this disinfectant is equally sufficient to get rid of any tomato brown rugose fruit virus particles in your greenhouse. But it seems to work because the eradication program was quite successful. Do not reuse rock wool or other substrates used in tomato production. There's only little experience available regarding the treatment of irrigation water. There are different methods available, such UV disinfection, filters, or heat treatment. They may reduce the virus load, but not eliminate. There is no data currently available that can show that tobamoviruses are completely destroyed using these technologies. When you look at the workers, it might be advisable to supply them with disposable coats, hats, and gloves for handling the plants in the greenhouse. And probably it's also a good idea to tell them not to eat tomatoes or peppers for lunch that have been brought in from supermarkets where we have no control from which countries these tomatoes or peppers are coming from, and they might be a potential source of infection. When we look at the affected area, so far no reoccurrence of the virus outbreak has occurred. So the disinfection management seems to have worked quite successfully. People are monitoring their plants and they are using randomized testing to detect a potential 1% infection in their crop. And they're using ELISA with the DSMZ antibodies and pool 25 samples. You can imagine if you have thousands or tens of thousands of plants, you need to cut it back to a sizable or manageable size for sampling. We had another suspected outbreak in a tomato breeding facility at the beginning of the year, but this could not be confirmed. But interestingly, we found something else, a new record for Germany, the southern tomato virus. And I spoke to Adrian last week, and he said that you also found this probably in the UK. It's not surprising because this southern tomato virus has a double-stranded RNA genome, but it's not known that there is an associated capsid or a shell. And therefore, this virus cannot be detected by electron microscopy, nor by ELISA or other serological tests. And the transmission rates by seeds are very high, up to 90%. And at the moment, no other vectors are known. And yeah, this virus seems to be a bit peculiar. Some say there is an asomatic infection of plants that have this virus. Others say that fruits can be smaller when compared to healthy plants. We have not, because this detection is so recent that we could not have done any experiments with this virus, but it has been recorded worldwide, including Canary Islands, Spain, France, Italy. Those are the European countries, but there are also reports from the United States and so on and so on. JKI again did a press PRA and decided it's not a quarantine organism. No eradication has to be made because the effect on plants, if at all, seems to be very minor. And then I come to my end. I just want to acknowledge my staff who have worked tremendously in the last few weeks on the diagnostics side of this virus. And yeah, thank you for your attention.
Thank you, Heiko. We've got lots of questions for you, if that's any, if you're happy to answer them.
Yep,
so,
that's fine.
One question, will the German fact sheet be made available for UK growers?
We are discussing to also translate into English, and it's an old publishing series from JKI that we are currently reviving, and therefore we had a lot issues with the layout and so on, but we are in the final proof stage, so hopefully the German version will be available next week or the week after, and then we can work on the translation. It will be open public access on our website.
Fantastic. A diagnostic question, or a couple of diagnostic questions.
Yeah.
So why did you use electron microscopy if you cannot use it to specifically identify the virus? I think that's, was it because it was an unknown?
For the samples that we get in at the time, it was an unknown virus, and tomatoes can be infected by 40 or 50 different viruses, and therefore we do not have the capacity to do 40 to 50 specific tests. But if we use electron microscopy as a first line of diagnostics, and we see whether there are rod-shaped particles, flexuous particles, or icosahedral particles, that narrows down the second line of testing, where we then can start with specific tests and use, for example, RT-PCR protocols. But it's a case-to-case decision that we make. If we get the question, is brown fruit rugose virus in these plants or not, of course we start straight away with the RT-PCR protocol.
Okay, so would the diagnostics allow you to rapidly detect the disease in transplants before they were dispatched to growers?
Should be. We haven't tested any young plants or seedlings yet, but I do think that the diagnostic protocols are sensitive enough even to detect the virus in low titer.
Okay, so it should be, but the test just needs some validation. How long does it take to get a PCR identification?
The if you use the generic primers, I think you can have the result within the day, but then you still do not know what type of tobamovirus is in your plant. And depending on we use a commercial sequencing provider, it takes another day or two to receive the sequences that need to be analyzed. And to be on the safe side, we say within a week you get a result and a confirmatory test.
Okay. And using your approach, could a lab come up with a wrong analytical result?
This virus is very difficult because of the risk of cross-contamination, and especially with the tobamoviruses, you always have a risk of cross-contamination. So if you have, for example, a smoker collecting the leaf samples with bare hands, you might have TMV on the plant sample that was not present in the plant, but you will of course detect that with RT-PCR. We are lucky that we have not had any cross-contamination in our laboratory as yet, but we are very, very careful when we handle these samples.
Okay, and also the sequencing should help give confidence.
Yes, that's why we say sequencing must be done to really identify what type of tobamovirus you get in your plant. The risk with the specific primers is that there might be variations of the virus around that might not be detected with this specific primer sequence, and therefore we prefer to use the generic Tobamovirus primers. The published sequences are very similar, so it doesn't matter if you have a virus from Jordan, from Israel, or from Germany. They are very, very similar, but I fear that if you have this, as I have shown in the one-step RT-PCR protocol, we did not understand why we got only 1 out of 3 samples positive, and then we decided to just work with the generic primers, full stop.
Okay. We've got loads and loads of questions, but in the interest of sticking to time for now, around the disinfectant.
Pardon?
So we've got lots of people asking if you can tell us what the disinfection is. We have lots of people asking if you can tell which disinfectant it is that's used in Germany.
It's called Menoflorades, and if you Google it, you should find their website. They have a couple of information on their website. This is their sheet, so there you find the name written in green, and they have done quite a few tests of the disinfectant and the efficiency against fungi, viruses, and bacteria, and they are currently the only licensed product in Germany. I know that there are some reports available that you can use skimmed milk or other products, soapy products, but none of them are licensed, and so we do not recommend using those disinfectants because we have no hard data supporting the efficiency of these products. And if you use skimmed milk powder, you do not have batch continuity. So please use only licensed products, and we do not have any experiments at the moment where alternatives have been tested.
Okay. Okay. We've got plenty of other questions around things like sources and quarantine status, but I think we'll probably come back to those once we've heard from Sharon, if that's okay.
Yeah.
So what I will do now is switch from Heiko to Sharon and let her introduce herself, and we'll go from there.
Right, good afternoon. I'm Sharon Matthews-Berry from Defra. Does that look okay? Is my screen? Can't see my presentation. Okay. Oh. Right, good afternoon. My name is Sharon Matthews-Berry and I'm from the Risk and Horizon Scanning team in Defra. And I'll talk a bit about the sort of regulatory aspects of this, but there also will be some overlap with some of the other presentations in terms of talking about distribution and the background to the pathogen. Okay, so as we've already heard, this virus was first described in the Middle East in 2015. It's closely related to tobacco mosaic virus and tomato mosaic virus. It was added to the UK Plant Health Risk Register following the first findings in Europe, which was the outbreak in Germany last year. Since then, the European and Mediterranean Plant Protection Organisation, EPO, have added it to their alert list. And for a sort of future, EPO are also planning to produce a pest risk assessment, and this will be developed later in 2019. And this will be a risk assessment for the whole EPO region for this virus. And I've just added at the bottom there a couple of links. This is the link to the UK Plant Health Risk Register entry and also to the EPO Global Database, where you can get some more information on the virus. So in terms of distribution, tomato brown Rugose fruit virus has a very scattered global distribution, and this means it may be present in other countries, but it has gone undetected. There also is the issue of the limited specificity of the ELISA test, and positive results may have been reported as tobacco mosaic virus. It was first described in 2015 in glasshouses in Jordan. It's since then been identified in Israel, where it's probably been present since 2014. It was found in Mexico in 2018. As you've heard about, the 6-7 outbreaks in Germany last year, and more recently, there's been outbreaks in Sicily and Italy. It's only 2 natural hosts have been identified of tomato brown Rugose fruit virus. This is tomatoes, and in Mexico it's been identified in chili peppers. There's been experimental inoculation carried out, which shows that symptoms are developed on varieties which have the resistance to tomato and tobacco mosaic virus, and also symptoms have developed on peppers which carry the resistance genes for Tobamoviruses. There are other experimental hosts which have been reported, Chenopodiums, Nicotinia, Petunia, and black nightshade. Just on this next slide, I was just going to talk a bit about the impacts that's been seen in different outbreaks. So firstly, the outbreaks in Israel in 2014. These were found in 6 mesh net houses, a total area of approximately 30 acres. The tomatoes there were seen to have mosaic leaf symptoms, which were described as mild to severe, and also occasional leaf narrowing. Fruit symptoms were seen on 10 to 15% of the total harvested fruit, and the affected fruit were yellow and described as being yellow and spotted. Experimentally, tomatoes carrying the resistant gene to tobacco and tomato mosaic virus were also found to be infected. And it was also noted there that the symptoms were not the same on all cultivars. In Jordan in 2015, this was again an outbreak in a glasshouse crop which showed 100% infection. The foliar symptoms were only considered to be mild, but there was strong brown Rugose symptoms on the fruit. To carry on with the impacts, there was also the outbreaks in Mexico, and this is obviously slightly different because this is not only tomatoes, but also chili peppers. Again, we're talking about mosaic symptoms and yellowing on the leaves, and fruit symptoms were seen on both crops, which were described as yellowing, green spots, grooves, irregular brown spots, and deformation of the fruit. To come on to the outbreaks in Europe, we've heard about the outbreaks in Germany already. the information I have is that we've seen foliar symptoms have been seen there. There's fruit with yellow spots and sometimes rugoous symptoms. I think from the initial outbreaks, we're talking about 10% of plants being affected in the glasshouse and showing symptoms. Recent information I've heard that from these outbreaks, the plants have been incinerated and the premises disinfected, and planting has resumed using tested plants, I've been told, and no further symptoms have been observed, and there's further testing planned. The more recent outbreak is in Sicily, in Italy. This is some quite new information I've got hold of only this morning. There's outbreaks at 5 fruit production nurseries, and then also at 2 nurseries producing young plants. From the original outbreak, it was described as being about 10% of plants infected. That was 600 of 6,000 were symptomatic. The symptoms were judged not to be severe, but some new information, which is what I've got this morning, is that a batch of infected seeds has been identified at one of the nurseries. So the seed has been tested, and the tomato brown rugouse fruit virus has been identified on it. And these seeds were described as being sourced from one batch from an EU member state and the other one from Peru. And as I mentioned before, the tomato brown rugouse fruit virus has been added to the UK Plant Health Risk Register. And in the next couple of slides, I'll go through the criteria which were considered in making that assessment. All right. Okay, sorry, I've just turned off the Adrian's just asked me to turn off the webcam because I've got a very slow connection. So I'll just go with the slides now. So when the risk register entry is assessed, there's different criteria that are looked at. The first one is the risk of entry. And so in this case, it was considered that the risk of entry would be due to either infected plants for planting or infected seeds. Now, as we've heard before, seed transmission has not been demonstrated, but it's quite strongly implied that it can be seed transmissible. It's known that other Tobamo mosaic viruses are seed-borne, including the closely related tobacco and tomato mosaic virus, and there's anecdotal suggestions that seed was the pathway of introduction into Mexico. There's also the new information that infected seed has been identified in Italy. We then look at the possibility for establishment in the UK. We don't see any reason why this wouldn't establish in the UK. The hosts are grown here, and as it's a protected crop, there is no issue of climate, so there's nothing that's likely to limit the possibility of establishment in the UK. So then we look at some evidence for spread. There's data on spread available from Israel. It was first detected in the country in October 2014. By February 2015, there are records scattered throughout the centre of Israel. By November 2016, their positive dissections are found in the extreme north. Now, the spread was considered to be human-aided, either by agronomists moving between sites and or by the trade in infected seeds or young plants. In terms of vectoring it, Tobamo mosaic viruses are usually considered not to need vectors, but there is some evidence that bumblebees can infect plants with tomato brown rugouse fruit virus, although it's unclear whether this is due to infected pollen or mechanical transmission. And the other criteria that we'd look at in terms of the risk register entry is the impact. And based on the current information concerning fruits, current information we have, there are the possibility of fruit symptoms that could render the infected fruit unmarketable. So also when producing a risk register entry, we would look at what current mitigations are in place. And this is what's in the currently in the legislation. So from our current legislation, we have that solanaceous plants for planting are prohibited from countries outside Europe and the Mediterranean. However, from the current known distribution, only Mexico is outside that European and Mediterranean area. So the prohibition against planting material is unlikely to have any impact on the movement of infected young plants. Also, this prohibition does not cover seeds. Seeds can be imported from any part of the range of the virus, but the requirement for seeds is that they must be acid extracted or have undergone equivalent measures. As a seed-borne Tobamomosaic virus, they are usually carried on the seed coat, though it's not known whether this applies to this virus, so acid extraction may reduce the chances of infected seed entering the UK. When a risk register entry is assessed, it also looks at uncertainties associated with assessment. And in this case, the key uncertainties would be, the first one would be seed-borne transmission not being demonstrated. In addition, there are uncertainties concerning the distribution. The full global distribution of the virus is likely to be greater than is currently known. In terms of hosts, are there additional naturally infected hosts out there? In particular, are there latently infected hosts which could be asymptomatic and act as a reservoir for infection? And then in terms of the pathways for entry, I think we come back to the seed-borne, the issue of the seed-borne pathway being unproven. And also, if it is seed-borne, does the acid extraction have any effect in reducing the rate of infection? So, possible future action. So this virus was discussed at the EU Plant Health Standing Committee last week, and there was support for developing emergency measures against it. It's considered that those emergency measures would focus on the input on seeds and the import of plants. The aim is to discuss the exact nature of these measures at the next Standing Committee in May, next month. So just to come on a bit to some possible options of what action would be taken if we did have an outbreak in the UK, it's considered that we would take statutory action against this virus. Therefore, action would be under plant health notice and supervised by the Plant Health and Seeds Inspectorate. I'm just putting together some ideas of the sort of measures we would take, but this is only really for guidance, because the exact measures would be considered on a case-by-case basis, taking into account the circumstances of the outbreak. It would be similar action to what we've taken in the past against potato spindle tuber viroid or calumnia latent viroid. So possible action would include the possibility of restriction on movement of fruit, but in terms of movement of fruit, that would only be restrictions of not allowing it to move to another production site for repacking. There would be we wouldn't envisage any restrictions on marketing of the fruit to wholesale or retail. It's just there is a possibility of spread from one production site to another via fruit. It also look at restrictions on movements of staff. So I think it was mentioned that in Germany there was a lot of spread about on sites, and so I think we would look at putting in place some requirements to prevent the movement of staff, for example, from an infected glasshouse into an uninfected one, and also putting in place hygiene protocols concerning wearing protective clothing, in particular gloves, and changing these when you move from visually infected plants to visually uninfected plants. And would also require cleaning and disinfection of tools to prevent movement between infected and uninfected houses and movement within infected glasshouses. The main control would be looking at a clean-up at the end of the season. We'd look at removal of plant debris. I think measures that we would definitely say would be fine for disposal of the plants would be burning, deep burial, and composting, so long as it was a PAS 100 standard composting site. But other methods could be considered in consultation with the PHSI, just to ensure they're appropriate. Other associated material would need to be removed and destroyed. For example, string, if it was biodegradable, it could go with the hulm. Rock wool, we would likely require that recycling for non-horticultural purposes. And then we would talk about the cleaning and disinfection of the glasshouse. And I think in addition to disinfectant, we would recommend cleaning with water and detergent to remove traces of organic matter, because this is something a point I'm often making, regardless of the crop, regardless of the pathogen, that disinfectants are often inactivated by the presence of organic matter. So it's very important to clean surfaces prior to disinfection. Another important requirement would be ensuring that any self-sown tomato seeds were prevented from germinating, to ensure that when you're introducing a new crop into the glasshouse, there's not old infected plants growing up anywhere. And additionally, once a new crop was introduced, there would need to be monitoring for the presence of these self-sown seeds, and additionally for symptoms within that new crop. Right, just really to conclude, this is a developing situation. There's obviously the potential for new EU emergency measures. There's also an EPO-PRA, which is going to be developed in the future. And because we're looking at something which we're not completely sure that we've found the whole distribution as well, it could be found in other places within the near future. And from a UK point of view, we are currently recommending that we would take statutory action, but this would be focusing on preventing the spread and cleaning up at the end of the season. So thank you very much for listening. So if there's any questions, I'll hand over to Adrian.
Thanks, Sharon. There are a few questions, and I think the first one will come down to some definitions. So, is tomato brown rugose fruit virus a quarantine pathogen in the UK?
As things currently stand, we would say we would take statutory action against it. So that would be under a plant health notice. So although it's not anywhere listed as a regulated organism, we would be treating it as such at the moment. Does that make sense?
Yeah, so I think it's worth pointing out that within the UK plant health regulations, that any disease that's deemed to be injurious to a plant is liable for action to be taken if it's deemed to be of a sufficient threat.
Yeah, there is within the legislation a line somewhere which mentions about plants not normally present within the territory, that you can take action on those. So there is, action would be taken under that piece of legislation.
So there's a few questions about what actions would be taken. So one, would plant health seeds inspector order total crop destruction?
I think that's highly unlikely, given the way we've dealt with similar viruses and viroids in the past. There's always the case that it's a case-by-case basis, and we would probably, I think to clean this up, you would be relying on having a crop break at some point, which I know for some growers is not their normal production methods. But in general, I don't think we would be requiring total crop destruction, no.
Okay. So, given the emergence of anecdotal reports in Holland, at the EU meeting, do you know if it was mentioned about a need for EU member states to inform other member states when an outbreak is present?
I don't know what exactly was mentioned concerning Holland and the Netherlands. I know that there are rumours that there have been outbreaks, and from an official point of view, I don't believe they have said there are no outbreaks. So I can't really comment on the situation in the Netherlands.
I think that's fair enough that we can only go with the official reports. Would you take measures against organic soil crops at the end of the season if infected? So that's probably thinking about risk of things like carryover in roots.
That's probably one of the sort of case-by-case situations. I haven't specifically considered organic crops, but yes, I do understand there that rather than growing in rock wool, you'd be in some kind of soil or compost. I think we'd have to consider what the risks would be associated with, for example, the roots being left behind, and probably be taking advice from Adrian on that one, I expect, amongst others.
Nicely passed, yes. So I think, given the time, I think there are a few questions to pass back to Heiko. If you're still with us, Heiko?
I'm still with you, yes. Can you still hear me?
I can still hear you. So within the German outbreaks, was there a time gap between the reporting from the different German crops?
No, I think they, within North Rhine-Westphalia, the different growers, they worked very closely together, and it was all detected at this very same time, basically. If that answers your question.
Yeah, I think that does. And within the German outbreaks and the disinfection advice, are you giving any specific advice on things like knife disinfection for de-leafing and things like that?
We can only give the general advice that's been given in the instructions from the commercial disinfectant, where they tested the times you need to successfully inactivate TMV. That's the only we can do, because we haven't done any experiments to test it with this virus, actually.
Okay. And I think that's part of the problem, that we're still in quite early days, and a lot of the work still needs to be done specifically on this virus.
Yeah.
Okay. I think, just checking through the questions, do you know what the perceived source of infections was in Germany?
Not out of my head. No, sorry, I can't answer you that.
Okay, that's fair enough. And again, I think as a plant pathologist, we always come in after the outbreak, don't we?
Yeah, and we also have the system in Germany that we have 16 federal states, and each federal state has their own plant protection organization that is dealing with the growers. And once there is a problem in the federal states, JKI comes into place, assisting in diagnostics, and JKI, a different department from mine, needs to be notified if it's a new notifiable disease. So we are basically the third in line when it comes to new outbreaks and confirmation of that. So yes, I regret a little bit that I cannot be hands-on in the greenhouses and look at the situations myself.
Do we know how the Israelis have dealt with the problem and how they are doing it now? So looking at different biocides, hygiene best practice?
I have
we just got the information that,
I'm afraid.
No, I think we've only got the information we've got to go on. Okay. There is a question about cross protection, and is anyone working on a mild strain for the virus?
Not that I'm aware of. What was found in Israel is very similar to what was found in Jordan, and again, what we found in Germany is also very similar to those two viruses. And at the moment, there is not much sequence difference. We only know that different tomato varieties have a different or differential behavior when it comes to symptoms. But we do not know if there's any mild strain available. And I have no knowledge if someone is intentionally mutating the virus to generate mild strains.
So again, I think that comes under, it's still very early days, isn't it?
Yeah.
As far as we can tell, everything's pretty much identical, so we don't have a mild strain to work with.
Yeah.
And so do we know which tomato varieties are more susceptible or less susceptible?
I think they're all susceptible, but the symptom development is different. On some, you get more leaf symptoms, and on others, you only observe the fruit symptoms. But in the end, we have not heard about a single resistant variety yet.
Okay. Well, I think I'll pick up the final question here, which is, can you tell us what work is going on in the UK at Fera, for instance? So I think I'm probably best placed to answer that question. So from our point of view, we have proposed some work to look at survival and disinfection for the virus. So we are hoping to look at a series of different disinfectants and to see how effective they are on disinfecting for the virus across different surfaces. And we're also hoping to look at survival of the virus on different surfaces and on hands, and just do some basic work on things like hand washing, so we can add into that hygiene best practice work. Okay, and I think that's us just about wrapped up for time. So Natalie, do you want to pick up and say anything to finish?
Just to finish, there have been some questions that haven't been answered. We've had loads through, so thank you for those. What I will do is try to collate an FAQ document and work with our speakers and team here to try and answer everything as best we can. And then I will publish that on the website alongside this webinar. So hopefully you'll get all the answers that we have at the moment, and if not, please email me, as I said, natalie.key@ahdb.org.uk, and I will do my best to answer your questions. Okay, well, thank you very much, everyone, and thank you to the speakers. Very much appreciated. And thank you to Adrian for hosting. You did a great job. Are you there still?
Yes.
Sorry, I was doing my classic of speaking on mute.
Okay. Yeah, well, I'll leave you to finish up then. Thank you.
Okay. So how do we finish the broadcast? I
can close it. It's just if you wanted to say anything else, that's all.
I think that's all we can say for now. As you say, we will collect the answers to the questions we haven't had time for. Thank you to everybody for listening, and thank you once again to our speakers. I think that's been a very useful way to spend an hour.
Yes, thank you very much. All right, bye.
Bye.